how do these bacteria prevent damage to the gastrointestinal tract? This is still a very new field, but I'd like to just talk about some of the hypotheses that have been raised. This is work that has been done by Dr. Andrew Nish and published in Science in 2000. And what he did was he took intestinal epithelial cells in culture and studied the mechanisms of how certain microbes can protect the gastrointestinal tract and others may hurt the gastrointestinal tract. And here is what he did looking at a pathogenic bacterium. Okay, this is a pathogenic Salmonella. And he found that the intracellular processes are different here with this pathogenic bacteria than with a non-pathogenic bacteria. The major difference is that the pathogenic bacteria breaks up this I-kappa B, the inhibitor of kappa B, so that NF-kappa B, the nuclear factor kappa B, can go into the nucleus of the cell, and this is a transcription factor that switches on the production of pro-inflammatory cytokines. The commensal bacteria, the good bacteria, prevent this from occurring, and they keep the I-kappa B together in a complex in the cytoplasm with the NF-kappa B so that the NF-kappa B cannot translocate to the nucleus and cause production of pro-inflammatory cytokines. So that's one hypothesis. Another hypothesis is that there are certain cells at the base of the villi here called the Paneth cells. And if you have normal resident microbes in the gastrointestinal tract, they have a crosstalk. They secrete substances that causes these Paneth cells down here to secrete certain enzymes. This is just angiogenin is one of these enzymes, which can kill exogenous microbes. Okay, another hypothesis. Another area that is a very exciting area that is being worked on by many scientists is the area of toll-like receptors. There are now approximately 11 toll-like receptors that are present on the surface of cells. These toll-like receptors bind to certain bacterial products. And here we see some of these toll-like receptors. This is toll-like receptor 4, and this binds to LPS. LPS comes from E. coli, pathogenic E. coli. Here you have TLR2, and that binds to gram-positive microorganisms. This looks like a very complicated process, and it is, but the binding of these toll-like receptors causes a signaling cascade that increases inflammatory cytokine production. Now we're beginning to find that it's important to have these toll-like receptors and to have some stimulation of these toll-like receptors, but when there is too much stimulation, that can cause pathology. Here is an example of some of this. This is, I don't want to get into too much detail with these studies, but this is a very important study that was published a couple of years ago from Yale University by Drs. Rakoff Nahum. And this was done in mice. And they looked at 4 different groups of mice, regular wild-type mice, and then they had these gene knockouts, MYD88. If you remember the previous picture, MYD88 is a linking molecule in the signaling cascade that links the toll-like receptors to the NF-kappa B system and that causes the production of inflammatory cytokines. Okay? They gave these mice oral dextran sodium sulfate to induce colonic injury. Now, DSS usually induces very mild colonic injury by itself. If you do something to exacerbate this, it can cause very severe injury. Well, one of the things that they did, they took these 4 groups of mice and they subjected them to the DSS, and they found that the mice that were the knockouts died, very rapidly died. Okay, if they did not have those toll-like receptors, those animals died very rapidly. Then they also depleted the mice of intestinal microflora. And they did another study where they reintroduced commensals and commensal products. The reason they did this was to see if getting rid of the microbes in the gastrointestinal tract actually would increase damage to the gastrointestinal tract. Okay? Now, how many of you use antibiotics in, let's say you have a small premature baby with respiratory distress? How many of you use antibiotics routinely in those babies? We do. Okay. Almost everybody, I know that probably everybody uses them routinely. Now, when I show you this data, begin to think about the routine use of antibiotics in our premature babies and what this might be doing. Okay. Question is, could these antibiotics have unforeseen or delayed consequences? Well, here's what they did. This group took these animals and gave them no antibiotics, the one group with no antibiotics, plus the chemical agent that injures the intestine. Then they gave them 4 different antibiotics. This is ampicillin, vancomycin, neomycin, and metronidazole, plus DSS. Then just 2 different antibiotics and 2 other antibiotics. And here we see that the group that was given the 4 different antibiotics, this is what happened to them. Their percent survival decreased very rapidly. So what happened to these animals was that the lack of microbes in their gastrointestinal tract made them very prone to intestinal injury by this chemical agent. Okay? So this is a cartoon that came from the New England Journal of Medicine that was published shortly after this article was published in Cell from the Yale group. And this shows that this is the normal situation here with intact toll-like receptor signaling and microbes present. You have normal protection of the GI tract. If there is a problem with the toll-like receptor signaling, you have enhanced sensitivity to injury and diminished capacity to induce repair. And if there are no microbes in the GI tract that are eliminated by the use of antibiotics, you have an enhanced sensitivity to injury and diminished capacity to repair. So the implications here is that the gut epithelium and the immune system do not simply tolerate commensal microorganisms, they're actually dependent on them. And manipulation of the intestinal microflora might lead to major consequences. Let's look into this some more. Well, in the neonatal intensive care unit, what is our top 10 list of medications that we use? Well, ampicillin and gentamicin. 95% of very low birth weight babies in our neonatal intensive care units get ampicillin and gentamicin. Now, why do we do this? We can't tell the difference between RDS and pneumonia. Premature delivery might be caused by infection. We want to prevent infections because of a high susceptibility due to immunologic immaturity. These babies have a lot of hardware in them, endotracheal tubes, OG tubes, umbilical lines, etc. And the sensitivity of blood cultures are suboptimal. So because of all of these factors, we use antibiotics very routinely in these babies. Now, why should we worry about this? Well, the first colonization of the intestine is one of the most profound immunological exposures in the newborn infant. As you know, the infant is born without microbes, but once those microbes establish themselves in the gastrointestinal tract, there's a huge amount of crosstalk that begins to occur between those microbes and the intestinal immune system that can last with us for an entire lifetime. Also, niches are formed as part of a potentially long-lasting biofilm, which is located in the luminal glycocalyx. This is just a picture of the biofilm formation. And it's one analogy of this is like if you have a garden, okay, and in that garden you have no plants growing, but you plant a weed into that garden, and that weed will eventually prevent the normal growth of other microorganisms or of other plants. In this case, it could prevent